Now, every molecule and ion in a fluid is in constant random motion from its own thermal energy. This is called Brownian motion. Now, the name comes from the observation that even inert particles suspended in fluid appear to almost jiggle erratically from being struck by surrounding molecules. Now, a concentration gradient is simply the difference in concentration between two regions. Diffusion is the net movement of particles from high concentration to low concentration. And I want to be precise about what net means here because it's a common point of confusion. Now, individual particles are moving randomly in every direction at all times, including from low to high concentration, just as often as from high to low, at the level of any single particle. However, because there are simply more particles in the high concentration region to begin with, more of them happen to randomly wander toward the low concentration side than in the reverse. The aggregate population level effect is net movement from high to low, even though no individual particle is trying to go anywhere. This continues until concentration is equal everywhere, and we call this equilibrium. Now, equilibrium would be when we have the same concentration of particles on both sides. Now, at equilibrium, particles are still moving randomly. The particles don't stop moving because they're in equilibrium. However, since concentration is now uniform, there is no more net movement. So the rate of diffusion depends on the steepness of the gradient, but also temperature and also the size of the diffusing molecule. So molecules that are smaller diffuse faster, and molecules that are hotter also diffuse faster. Now, when diffusion happens across a membrane specifically, an additional factor kicks in. Can the substance actually get through the bilayer at all? Simple diffusion means crossing directly through the lipid bilayer with no protein assistance. It is driven purely by the concentration gradient and requires no energy input. Now, this is why we classify it as passive transport. Only small, nonpolar, uncharged molecules can diffuse through a membrane. Now, the clearest examples that we have of these small, nonpolar, uncharged molecules are respiratory gases like oxygen and carbon dioxide. This is why neurons don't need any specialized machinery to bring in oxygen for cellular respiration or to expel carbon dioxide waste. These gases simply diffuse directly across the membrane on their own gradient. Now, large polar or charged substances cannot cross this way at any meaningful biological rate. Now, a charged particle trying to enter the hydrophobic core is chemically like trying to force the wrong ends of two magnets together. This includes sodium, potassium, calcium, and chloride. Now, these four molecules in particular are going to be really important when we start talking about neuronal communication. So when we actually start getting to neurophysiology, we really need to pay attention to sodium, potassium, calcium, and chloride. Now, all of these guys are surrounded by a shell of water because they are charged. So these ions require dedicated integral membrane proteins that we call ion channels to cross them. Now, osmosis is simply diffusion where the diffusing substance is water. In this case, it would be water moving across a selectively permeable membrane. But what does that mean, selectively permeable membrane? That just means it's a membrane that lets some things through freely while restricting others. Now, osmosis is the net movement of water from a region of higher water concentration to lower water concentration. Equivalently, and also a little bit more intuitively, water moves from lower solute concentration to higher solute concentration. So it's the same statement, basically. We're trying to get to the same point. More solute particles occupying a given volume means fewer water molecules can occupy that same volume. So higher solute concentration is equivalent to lower water concentration. Now, cell membranes are naturally selectively permeable in exactly the way this requires, right? So water is small and polar, and despite its polarity, it's small enough and concentrated enough on both sides that it can cross the bilayer to a meaningful degree by simple diffusion. And many cells also have specialized proteins called aquaporins. Now, these are dedicated water channels that dramatically increase the speed or the rate at which water is moving without changing direction. The direction the water is moving is still set entirely by the gradient itself. Now, something that helps me remember this is I had a microbiology professor in college who used to say water parties with the salts. So water always wants to go where there are more solids or more solutes. Osmotic pressure is the amount of mechanical pressure that you would need to apply to a solution to stop net osmotic water flow into it. Now, osmotic pressure is a property of the solution, so it's describing its tendency to pull water in. It is not a description of the membrane, and it is not a force actually being applied under normal conditions. This is a theoretical force. Now, higher solute concentration means higher osmotic pressure. Tonicity describes the effect a surrounding solution has on cell volume. That is based on the concentration of the non-permeating solutes outside relative to inside. Now, isotonic means equal concentration, so water moves in both directions equally, so there's no net volume change between the two sides. Hypotonic would be a lower solute outside than inside, so the water concentration is higher outside. So net water movement is happening into the cell. So more water is coming into the cell in a hypotonic solution. Now this causes swelling. And if the swelling becomes severe enough, lysis. Now lysis means to burst. So in this image, we have a red blood cell, and you can see that like biconcave disc here, which is characteristic of a red blood cell. So you can see in our isotonic solution, the cell is perfectly sized. We have a pretty even number of solute and water concentrations, both inside and outside of the cell. So this is perfect condition. Now, hypotonic solution, you see the cell gets really large and it's burst. So we've got two areas where the cell, it's become too large, the cells burst to release those water molecules. Now, something I use to remember this is the hypotonic. That O, I think of the O being what the cell looks like when it's swelling. So hypotonic, swelling like the O. Hypertonic would indicate a higher solute on the outside. So that means we have net water movement out. And if we're losing water, it's going to cause the cell to shrink. Now, in this example here in the red blood cells, we would call that crenation. Now, this is when the surface takes on this really like shriveled, scallop looking appearance. I want to talk about this now in the context of neurology. Astrocytes, which we will talk about later, actively regulate the ionic and osmotic environment around neurons. And disruptions to that regulation, sometimes from injury or disease or from extreme fluid imbalance, can cause neurons or glial cells to swell or shrink in ways that directly impair neural function. So this concept of tonicity is very important when it comes to neuron health. Now, something I use to remember hypertonic shrinking is I usually think if someone is really hyper, they're moving around a lot, they tend to be really skinny. But that's, a generalization. But if you're moving around a bunch, if you're really hyper, I think of somebody really tall and skinny in life. Now, we've established that charged ions like sodium, potassium, calcium, and chloride cannot cross the bilayer by simple diffusion. Since neurons depend almost entirely on controlled movement of these ions, we need a mechanism that lets them cross despite the hydrophobic core. Now, that's something we call facilitated diffusion. So it's the passive movement down a concentration gradient made possible by an integral membrane protein that provides a route through the bilayer. Now, it's still passive transport in every sense that matters. The net movement is always high to low, and it requires no cellular energy. The only thing that we've added at this point over simple diffusion is specificity. So a specific protein for a specific substance, and speed for that matter. And this is particularly critical for ions that essentially cannot cross unassisted at any meaningful rate. Now, what do I mean by specificity? Some of these integral membrane proteins are not very specific, so they can let a bunch of different molecules or ions in and out of the cell relatively easily. However, some of these integral proteins are very specific, so it generally only lets a specific ion or a specific molecule through the membrane space. Now, the first family that we'll talk about that's carrying out these facilitated diffusions is channel proteins. Now, these are multi-pass integral proteins. Let's break that down. Multi-pass meaning that the protein itself passes through the membrane multiple times. Integral meaning it's embedded into the membrane. And these multipass integral proteins, these have transmembrane alpha helices that arrange around a central water-filled pore running straight through the membrane. Now, the pore's interior is lined with hydrophilic chains. This makes sense because we've got water and all sorts of ions and other molecules that are hydrophilic because they can't pass through the membrane, passing through the inside. So we want like to like, hydrophilic to hydrophilic. So the interior will be hydrophilic. Unlike the outward-facing surface of those same helices which contacts the lipids. So the outside of the protein, where it's actually embedded into the membrane, will be hydrophobic. And so we've got water and appropriately sized or charged solutes passing through without ever contacting the hydrophobic environment. Now, because the pore is essentially a continuous open tunnel in the open state, ions move through extremely fast and often approaching the theoretical maximum rate for an ion diffusing freely in water. Now, specificity comes largely from the selectivity filter, which is the pore's narrowest point, where precise dimensions and precisely arranged charged or polar side chains interact with an ion's charge and its water shell in a way that favors one species over another. And we'll come back to the mechanistic detail of these selectivity channels in a later session. Now, many of these channels aren't simply opening all the time. They switch between open and closed states, which is a process called gating. And it's classified by what triggers it. So a ligand-gated channel opens or closes when a specific chemical messenger binds a site on the protein, which triggers a conformational change mechanically transmitted to the pore. A voltage-gated channel responds to a change in the electrical voltage across a membrane, which is sensed by a voltage-sensing domain. These are going to become incredibly important when we start talking about action potentials. Now, a mechanically gated channel opens in response to physical force, so a stretch, pressure, or vibration, which is especially important when we get to touch and to hearing, which we'll cover later. And then last we have leak channels that lack a strong gating altogether. These stay open or nearly open essentially all of the time and allow a steady trickle of ion movement down the gradient. Now, potassium leak channels are specifically very important because they play a major role in establishing a neuron's resting electrical state, which we'll cover in detail soon. The second family is carrier proteins, also called transporters, and these are mechanistically distinct from the channels in a fundamental way. Now, rather than forming a continuous open pore, a carrier binds its substance on one side of the membrane, undergoes a conformational change that exposes the bound substance to the opposite side, and then releases it there. At no point during that cycle is there an open pathway connecting both sides simultaneously. That is the key structural distinction from the channel. So channels, open all the time. Carrier proteins or transporters, open, close, open and release. Now, the classic example of these transporters or carrier proteins in neurology is the GLUT family, the GLUT family. These are glucose transporters. Now, these bind glucose on the higher concentration side, then they undergo a conformational change and release it on the lower concentration side. Now, a channel's transport rate scales roughly linearly with the gradient. So a carrier's rate increases with the gradient only up to a certain point and then kind of plateaus. We call this plateau saturation. Because there's a fixed number of carrier molecules, and once every single one of them is continuously cycling as fast as it structurally can, no additional gradient steepness can increase that rate any further. Now, if you'll recall from earlier that net diffusion always precedes high to low concentration because that direction is thermodynamically favorable. It's spontaneous and requires no energy input. Moving a substance in the opposite direction, low to high, works against that natural tendency. It is thermodynamically unfavorable and will not happen spontaneously. Exactly like it takes energy to push a ball up a hill rather than to let it roll down. It takes energy to push an ion or molecule against its gradient, and that energy has to come from somewhere. Now, active transport is defined as movement against a concentration gradient accomplished by an integral protein that couples this unfavorable movement to some kind of energy source. Now, primary active transport means the energy comes directly from a chemical reaction inside the transport protein itself. It almost always happens because of the hydrolysis of ATP, which is adenosine triphosphate. Now, ATP is the cell's principal short-term energy currency. ATP is built from adenine, a ribose sugar, and a chain of 3 phosphate groups. Now, the bonds linking those 3 phosphates are relatively unstable and release substantial usable energy when broken. Now, when an enzyme removes the outermost phosphate, so that 3rd phosphate on the end, through hydrolysis, ATP, or adenine triphosphate, becomes ADP, or adenine diphosphate, and the released energy becomes available to do cellular work. Now, a protein that carries this hydrolysis process out and uses that energy to power some other process is called an ATPase. Now, we know that ending ase, ASE, refers to some kind of enzyme. So it's an ATP enzyme, ATPase. And it is a membrane transport ATPase that uses the energy specifically to move a substance against its gradient. And that is the defining mechanism of that primary active transport. Now, for a specific example, the single most important ATPase for our purposes is the sodium-potassium ATPase, also called the sodium-potassium pump. Now, it's found in the plasma membrane of essentially every cell in the body, including every neuron. It moves 2 ions simultaneously, both against their gradients. It moves sodium out of the cell from an area of lower concentration inside to higher concentration outside. It also moves potassium into the cell from an area of lower concentration outside the cell to higher concentration inside the cell. Now, this pump is precisely what creates and continuously maintains those 2 gradients against their natural tendency to dissipate through leak channels. The stoichiometry is fixed and precise. So all that means is the number of sodiums and the number of potassiums being moved is the same every time. For every single ATP hydrolyzed, exactly 3 sodium ions move out and exactly 2 potassium ions move in. Now, 3 positive charges are leaving. Those potassium ions are positively charged. So we have 3 positive charges leaving and 2 positive charges entering. Those sodiums are also positively charged. So when we bring 2 in, we have a net gain of 2, or we have 2 positive charges entering because of the sodium. Now, if we have 3 leaving and 1 coming in, it means we're losing 1 net positive charge every cycle. So this makes the pump electrogenic. Now, what electrogenic means is that it directly contributes to the cell's electrical charge difference on top of its larger role of building that concentration gradient. So now I'm going to walk you through the actual mechanical cycle. Now, the pump alternates between 2 conformational states, E1 and E2. Now, in E1, binding sites face the cell interior with a high affinity for sodium. So then we have 3 intracellular sodium ions binding to the inside. Now, when those 3 sodium ions bind, it triggers the hydrolysis of 1 ATP molecule, which removes energy, or it releases energy. Now, when we hydrolyze that ATP molecule, we remove 1 phosphate group from the ATP. That phosphate group attaches directly onto the pump protein, and we call that attachment of that phosphate group phosphorylation. Now, this phosphorylation event drives the major conformational change, converting the pump from E1 into E2, where the binding sites flip to face the extracellular side, simultaneously switching affinity. So when we were facing the inside, the protein really wanted to bind to sodium. Now it doesn't. Now it really wants to bind to potassium. Now, because E2 no longer holds sodium tightly, those 3 sodium ions get released into the extracellular space, so outside the cell, and then 2 extracellular, so outside potassium ions, immediately bind to the now vacant sites. Now, those potassium binding to the protein triggers the release of that attached phosphate. So that phosphate that we just took from that ATP before, it now releases. We call that process dephosphorylation. And losing that phosphate reverts the protein back to E1. So it flips the sites inward again, and now we want sodium once more. Now, because we're favoring sodium, we release 2 potassium ions back into the cytoplasm, and that is 1 full cycle of that sodium-potassium ATPase complete. Now, this is a phosphorylation-driven cycle, which is why the sodium-potassium ATPase belongs to a broader class called P-type ATPases. P standing for phosphorylated. So all of these pumps that are P-types will be phosphorylated at some point as an intermediate step. Now, because this pump runs continuously in essentially every cell in the body, it consumes a substantial fraction of the body's total ATP output. And neurons specifically, which are constantly restoring the sodium and potassium gradients they use up during electrical signaling, running this pump is one of the single largest energy expenditures in the entire nervous system. This is not a minor housekeeping cost. It is one of the most expensive things a neuron does, and it happens continuously because it has to remain ready to fire. Now, the sodium-potassium pump that we just covered builds up a steep sodium gradient with a great deal of stored potential energy in it. The secondary active transport moves a substance against its own gradient, but the transport protein never hydrolyzes ATP directly. So, in the next lesson, we'll start talking about how it is powered through a secondary substance.